All right, guys, thanks for joining me here for week 3. Nice job for sticking with it. This is lesson 5, working on how Bill Gates knew that that a pandemic was coming. So let's start with the learning targets for today. So two things that we need to review today. Number one, how is a protein made and how is it coded for? So this is a review from what we did in the fall. And then number two, um, how have the virus spikes, those proteins that are on the outside of the coronavirus, how have they been able to change in order to infect humans? So we actually have some really interesting data to look at of the actual protein on the outside of the virus that we're all dealing with right now, and where we can be able to see how it's changing over time. At the end of today, you're going to have an assignment 5 to complete and a Google form. So before we get going, let's review what we know so far 2 weeks into this. Number one, we know that all traits in organisms exist for one of 3 reasons. Either number one, they exist to help the organism get energy and nutrients. That's like the whales hunting the fish. Number two, they exist to avoid death, like the octopus. Number three, they may exist to make copies of itself, like birds that have colorful feathers. That's true of everything. Animals, bacteria, humans, it doesn't matter. All traits are based on those 3 things. Second thing we know, no 2 individuals are exactly the same. So even birds of the same species could have different sized beaks, just like people are different size heights. And so those variations could have a positive or negative impact on one of these 3 things up here. Third thing we know, viruses, the only way that they can enter a host is by matching the protein on the outside of their membrane with the proteins on the outside of the host membrane. If it doesn't match, it can't enter the cell. Number 4, something called antigenic drift and antigenic shift are things that lead to variation in those proteins on the outside of the virus. So over time, the proteins on the outside of viruses are always changing. In some cases, they change slowly over time, like in this case here. In other cases, they change really fast all at once. Number 5, virus proteins that match the proteins of a new host will be able to make more copies of itself faster than viruses that cannot. So what I mean by that is the better the match is between the proteins here on the outside of the virus to the proteins on the outside of the host, the better that match is, the faster they can replicate and the better they're going to be at making copies of themselves. Number 6, we also know from the last lesson that SARS-CoV-2, which is what we're dealing with right now, that's the coronavirus that's causing the pandemic, likely started in bats, because that's where we see almost all coronaviruses. But then somehow that particular coronavirus, SARS-CoV-2, changed in a way that allowed it to infect humans. So it went from having a bat host to a human host. Okay. So now that we got that, here's the question for today that this lesson is going to be about. So if it started in bats right here, how did the virus on the outside, how did the virus change in order to be able to infect human cells instead? What specifically happened to allow that jump to occur? So as we start thinking about that question, let's remember that if a virus is going to infect a new cell, the proteins on the outside, which we call spike proteins, need to be able to match with the receptor proteins, those are these ones here, on the host. So if it started off looking like this, it's going to have to change in some way for it to be able to infect the human cell. Just to review here, proteins over here are made up of these little circles right here, which are amino acids. And there's 20 different amino acids, and the way that we know what order or which amino acids to put in there to make a protein like this is that information is coded for by DNA, or in the case of a coronavirus, by RNA. So DNA and RNA hold the instructions for making proteins. DNA is composed of 4 different nucleotides, A, T, C, and G, and those 4 letters are what are used to instruct the cell to make a protein. Proteins are made up of amino acids. There's 20 different amino acids, and if you put them all together, it makes a big long string of amino acids together, and that string then folds and makes a shape like this. And it's the shape that, of course, will determine if the virus can infect the host. So check this out. This is the genome of a coronavirus, very similar to the virus that we have going around in humans today. So we know that coronaviruses start in bats, right? And a coronavirus has an RNA molecule in it full of the A, T, C's, and G's that in this case is about 29,000 letters long. So those 29,000 letters code for the entire virus. Within there, there's a section, this section right here, that codes for the spike protein on the outside. And if we look closer, down here at the bottom, this is the list of amino acids that if you were to put those together, that would make a spike on the outside of the virus that would allow it to infect bats. But this same spike, if you were to match it up with a human host, it would not be able to infect the human host because it's the wrong shape. So if we compare that to the spike protein that we have today in SARS-CoV-2, which is the virus that's infecting everyone today, let's compare that. So we have data today on what the amino acids are in the spike on the outside of the SARS-CoV-2 virus, the one that's infecting everybody today. And most of the letters are the same. Okay, so the amino acid sequence of the bat spike protein is pretty similar to the human protein, except for these 1, 2, 3, 4, 5, 6 places, everywhere where there's a blue square. Those blue squares are the key for allowing this virus to infect a human. If you change the amino acid in those 6 places, then all of a sudden, boom, we go from a virus that is able to only infect bats to a virus now that can infect humans. Okay? So if it only takes 6 specific amino acids, if you only need to change 6 of them for it to be able to infect humans, and these coronaviruses are happening in bats all the time, that's kind of scary. I wonder how often those amino acids change. So we've been taking samples of the virus, of the coronavirus, the SARS-CoV-2 virus that's going around in people. We've been taking samples of that for the last 3 months since December, or 4 months since December, and we've been looking at the A, T, C's, and G's in the genome and trying to figure out how often this virus is changing. And every dot on here is a different variation of the virus that's going around. So many people around the world have gotten the virus. Hundreds of thousands of people have gotten sick by this coronavirus. And when we look at the actual virus, we realize that these viruses that people are getting infected by are not all the same. In fact, if you start with one virus, after about 2 weeks, it changes in some way. One of the amino acids changes. Okay, and every 2 weeks it does that. So we start off with 1, and then after 2 weeks we've got 2 different strains of the virus, and then after 4 weeks we've got 4 strains, then 8, then 16. And that means that today, after 4 months, around the world we have about 250 different variations of the SARS-CoV-2 virus. So they can all infect people, but if we look at the amino acids, they're all slightly different. There's 250 different versions in the world. Okay, that was kind of a lot. If you need to go back and listen to that again, you can, and then click on the Google form and see if you can answer those all correctly. That'll help you sort of narrow down what I want you to focus on. And then I want you to go to assignment 5 and think about what are the chances that if we only need to change 6 amino acids, what are the chances that that would happen? Like, how easy is it for a virus to jump from a bat to a human? Okay, nice job, guys.